Hello student, today we are going to discuss about the various in vitro culture techniques under the plant tissue culture topic. This topic is particularly for the second semester BT 1 to 4 course and the degree course is BTech Biotechnology under VNMKV Parbai. Now first of all of you are well aware of the plant tissue culture. Plant tissue culture is nothing but a culture of whole plant part or what we say a plant under in vitro culture, under in vitro condition, which is aseptically maintained for the regeneration of whole plant or plant part. Now there are two aspects which are more important. One aspect is what is in vitro. Okay, now we look at what is in vitro. In vitro, it's something that means inside a glass, or what we say, the condition where everything is kept under control condition inside a laboratory. We call it as a in vitro condition. Okay, and then there is again another word, that is sterile condition. Sterile condition means a condition where there is a absence of microorganism. Sterile condition is a condition where there is absence of microorganism. Now for the sake of tissue culture, there are different types of tissue cultures. Okay, we have actually calculated 12 different types of cultures, okay, which happens under in vitro condition aseptically. Number one is a seed culture. Okay, so as you all are aware of, seed is nothing but the encapsulated form of embryo. Seed is nothing but the encapsulated form of embryo. So now seed culture refers to culturing the seed under in vitro conditions. So what we have to do, we have to obtain a seed, we have to sterilize it, and then we have to culture it on a suitable culture medium under in vitro condition. Ultimately the seed will give rise to the whole plant. This technique is particularly useful for those plants whose seeds are fail to germinate naturally, or they have large problem for what we say abortion of seeds. Okay, so we call it as a seed culture, simply culturing a seed under in vitro condition. Okay, now there is organ culture. So which plant part we are using for tissue culture, we refer to it as a organ culture. Now if we use root, then we call it as a root culture. If we use leaf, then we call it as a leaf culture. If we use node, then we will call it as a node culture. Okay, if we use shoot tip, then we call it as a shoot tip culture. So there are different types of plant. If we look at this plants has various part. Now the plant part that we are actually selecting for the tissue culture is known as a explant. So what we have to file, we have to discover for the mother plant. Okay, mother plant should have very peculiar characters. Okay, and from that mother plant we collect its part for culturing under in vitro condition. Now it should be remembered that the part of mother plant we are using for tissue culture is known as explant. Why we call it as a explant? Because initially it was a part of mother plant, but now we are removing that particular part from mother and now we are culturing it under in vitro condition. So we call it as a explant because it was a part of plant. Now before culturing the explant, we need to sterilize that explant and then we need to move for the further culture. Okay, now what part we are using, we call it as if we are using shoot tip, we call it as a shoot tip culture. We are using root, it is called as root culture. We are using leaf, it is called as leaf culture, and it is also called as nodal culture. Now then there is the next type, protoplast culture. Now if we look at the protoplast, so protoplast are nothing but the cell without cell wall. Now we know that all plant cells had cell wall. Okay, so if we remove this cell wall, okay, if we remove this cell wall, then there is inside a plasma membrane. So this plasma membrane cultures the cell or binds the cell. So cell without cell wall is known as Protoplast culture has its own application. Okay, now the cell without cell wall is known as protoplast, and if we culture this protoplast under in vitro condition, it is known as protoplast culture. Then there is a callus culture. So callus is nothing but the unorganized mass of cells or unorganized mass of cells. This unorganized mass of cells is formed under in vitro condition, and it is known as a callus. So if we are culturing this callus, it is known as a callus culture. Okay, and callus culture has many more applications. Okay, so then there is another anther culture. In anther culture, we isolate the anthers which are definitely haploid. We call it as an anther culture. In between there is a suspension culture. Suspension culture actually stands for the term of liquid media. In all other cases, we can easily use solid media. If all these cultures are happening under liquid condition, it may be protoplast, callus, seed, organ, ovule, ovary, all these culture if happens under liquid medium, we call it as a simply suspension culture. Okay, if we use anther culture, we isolate the anther from the plant, we culture it under in vitro condition, and it is haploid anther, so we call it as a anther culture. So ultimate aim of anther culture is generation of haploid plant through the process of androgenesis. Okay, androgenesis means formation of haploid from anther or pollen is nothing but a androgenesis. Similarly, there is a pollen culture. So pollens are inside anther, they are haploid. If we are culturing pollen, then it is known as a pollen culture. Okay, if we use female plant parts, ovules, okay, then it is known as a ovule culture. Then there is also ovary culture. If ovules are unfertilized, then they will lead to the formation of what we say as a haploid plants. If they are fertilized, then they will lead to the formation of what we say diploid plants. Ovary can be also cultured, and it will lead to the formation of haploid plants. If we culture ovary, it is known as ovary culture. If we culture ovule, it is known as ovule culture. Okay, then there is endosperm. We all know that there is a presence of endosperm inside a fertilized ovule. Endosperm is generally triploid tissue, so formation of triploid plant is possible from the endosperm culture. Then there is a embryo culture. Embryo is a diploid structure, so culturing embryo will lead to the formation of whole plantlets. Okay, and then there is a finally somatic embryogenesis. Somatic embryogenesis or somatic embryo culture is a process where we used to form a we used to have a diploid cell, any plant diploid cell. We culture it under in vitro condition, and finally it will lead to the formation of embryoid-like structure, that is a somatic embryo, because embryo is formed from somatic cell is known as a somatic embryo, and this somatic embryo will finally lead to the generation of whole plantlet. In this manner, there are different types of cultures. Now please remember that when we use any plant part or any plant cells under in vitro condition, it may either undergo the what we say organogenesis, which lead to the formation of different organs just like shoot, root, or it may lead to the what we say formation of callus, so which is undifferentiated mass of cells, or this callus then further subjected to the collagenesis, that is formation of shoot, rhizogenesis, that is formation of root, or colorhizogenesis, that is formation of both shoot and root at a time. Then these plant cells may lead to the formation of somatic embryogenesis. Somatic embryo is a concept. We know that embryo is formed by the fusion of male and female gamete. There is a formation of zygote. When zygote divides, then it leads to the formation of embryo. But in case of tissue culture, these somatic cells, when we culture them under in vitro condition, they will lead to the formation of embryo, and this embryo is formed from somatic cells, so we call it as a somatic embryo, and it behaves like a gametic embryo, okay, and lead to the regeneration of whole plantlets. Now, then there is also formation of what we say cytodifferentiation, which lead to the formation of xylem and phloem. In this manner, there are different types of tissue culture techniques, which means culturing various plant parts under in vitro condition for the regeneration of whole plants or plant part. Okay, so now in this topic we have covered different types of tissue cultures. Okay, thank you very much.